Hi, everyone. Welcome back. I want us to be able to get through the material and make good progress with the class. So for that, we're going to do a pre-recorded lecture today, just to make sure we get through what we need to get through. Let me share this screen via this. Okay, great. Yeah, obviously I sent I posted on Canvas to keep you guys in the loop of how Bruna and I think you're doing in the course. Obviously the lab exam grades were very disappointing. I'm not going to go into that a lot right here because I wrote all that I really needed to say in that announcement on Canvas. But we still have a bunch of semester left. And so let's just try to stay positive and get through the material and learn the material. And so for those students who are doing very well, keep it up. Whatever you're doing is working, so that's great. For those of you that are doing okay, but wish you were doing a little bit better, there's still time to kind of adjust. For those of you who are consistently doing poorly, you really need to spend more time with the material. And several of you have written to me for office hours for this coming Thursday, such that I don't know if I'll be able to accommodate everybody that day, but I will be getting back to you sometime today, Tuesday, to confirm about office hours. All right, so we're going to leave that to the side and we're going to focus on new material, right? New material. So we're finally, believe it or not, to the seed. I know, it's exciting. And today we're going to talk a little bit about the seed, in that we're going to talk about the ovule. Okay. So we're going to introduce seed plants and we're going to talk about the amazing ovule. Oh, I can't even say it. Let's try that again. The amazing ovule. All right, so where are we in the plant kingdom? We have come a long way, very long way. So here are all of the divisions that we cover in this course. Long ago, feels like, we covered the non-vascular plants, right? The mosses, liverworts, and hornworts. And then with the ferns, we finished up those spore shedding vascular plants. And so now we're done with those. We're going to keep moving on through the rest of the vascular plants, which are the seed plants. And the seed plants, there's two different types. There's something called an angiosperm, which we'll cover at the end of the semester, and something called gymnosperms, which we'll start discussing in more detail on Thursday. So this might seem familiar because we talked about this a lot toward the beginning of the course. Seeds are a big deal. They are a significant milestone for plants and for us as people who eat things from plants, a lot of times seeds. So you could see here on this slide are just some examples of the seeds we actually eat. Yes, and so obviously seeds are a big evolutionary milestone for plants. Part of the reason we know that is because the majority of plants on Earth make seeds, right? Especially the flowering plants, which are those angiosperms that we'll talk about at the end of the semester. And so the majority of plants on Earth are seed plants, particularly flowering plants. And so obviously that's a system that works very well. Having a seed works very well. That's why the majority of plants on Earth have seeds. So, but what are other ways that seeds give plants this kind of evolutionary advantage, right? Like, what's good about having a seed as versus not having a seed? Because as we've already seen in this course, there's plenty of plants that don't have seeds, right? There's mosses, liverworts, hornworts, lycopods, ferns, horsetails. None of those have seeds, and they do fine. But what are some reasons that seeds might really be helpful? So one reason is that seeds have what's great survival value. So they improve the ability of the plants to survive and to make new plants. So seeds provide better protection for the embryo, and they provide a food source for the embryo, which we have not seen so far in this class, right? So it's like the seed is almost like there's like a little lunchbox in there for the baby plant. And so seed plants have a selective advantage over plants that shed their spores because the embryo is better protected and there's a food source for the embryo. If we were to look at the history of plants on Earth, right, we've seen this slide before, back in the day. But if you look here, so the first seed plants show up around, just under 400 million years ago. Right, and those are your ginkgos, conifers, and cycads, meaning the ones that are still around today. Those are the ones that are still around today that showed up back then. And if we were to compare what's going on with the animals around that time in the fossil record, you can see, look, when did this say? First seed plants appear about 380, 360 million years ago, right? So 380, 360. So if we look here, and I guess black will be a good color on this slide because this slide has a lot of colors. Black will stand out. 360, 380, right? So you're looking at like right around here is when the first seed plants arrive or show up in the fossil record. And you can see before then, what are you looking at? You're looking at fishes, you're looking at corals, things that live in the water, right? Obviously, we already learned that as plants start to dominate the land, so too do animals dominate the land, right? We see the first amphibians showing up. We see insects showing up. But that 360, 380 million years ago, seed plants show up, and then look how much diversification we see in animals after that. Because as plants become more diverse, so do the animals become more diverse as well, because there's like a feedback system of who is eating who and things like that. So the evolution of the seed leads to a diversification event in the animals. See that at the bottom? Okay. Right. This is what I was just drawing. Right. Okay. So far in this class, we've really focused on the spore shedding plants, right? These are those phyla that we have covered so far, the non-vascular and the vascular phyla that we have covered so far. Notice all of those words on the left end with phyta, which is how you know that it is a phylum, right? But look at this through like evolutionary time, and I think black still works well here. So look, here's our earliest plants, right? The mosses, liverworts, and hornworts. And all of these are homosporous. Then we get to the vascular plants, and we see a little bit of both, right? So like the lycopodium lycopods are homosporous. Equisetum is also homosporous. Most of the ferns, particularly the terrestrial ferns or those in Phyllocales, remember we discussed the orders of ferns before the break. So if Phyllocales doesn't sound familiar to you, you should go back and watch that lecture and look at those slides. So we still see some homosporous sporophyte plants, but we start with Selaginella, we see a big change in that now sporophyte plants are going to shed two different types of spores, right? They're going to be heterosporous sporophyte plants. And we saw that also in the aquatic ferns, right? That were also, remember Marceliales and Salviniales? That was also in that fern orders class. But if you look at a general trend going from earliest plants to later plants, you see a shift, right? Homosporous, homosporous, a little bit of heterospory, homosporous, and then heterosporous again, right? We're shifting from blue to red there. And what do you know? When you get to the seed shedding plants, gymnosperms and angiosperms, they are also heterosporous. So going forward, see, All the rest of the plants that we're going to cover in this course, which are seed plants, are heterosporous. So no more homosporous. Everything we're going to cover, all the sporophytes that we cover now are going to have two types of spores and be heterosporous. They're going to make megaspores that will give rise to the female gametophyte plant. And they're going to have microspores or make microspores that give rise to the male gametophyte plant. And if you look at your book, if any of you remember that I gave you the textbook at the beginning of the semester for free, and it's available on Canvas. So if you're looking in your book, you might see the terms megagametophyte and microgametophyte, which just mean the female gametophyte and male gametophyte plant respectively. And you might remember, trying to remember which lecture it was. It was the lecture that it kind of was a review lecture, kind of re-examining the shift of the differences in generation, like going from plants that the gametophyte generation is dominant to going to the plants where the sporophyte generation is dominant. And one thing we talked about in that review class was like how seeds are actually, how this heterosporousness, right, is ultimately going to give rise the seed. It may have actually been in the Selaginella lecture. I can't remember right now which one it was, but we did cover it. But so seed plants are all heterosporous, and the seed itself is actually results from this extreme form of heterospory, in that we have this ovule, which after is fertilized will become the seed. So what is an ovule? Maybe you've heard the term ovule before, right? Humans have ovules as well, right? Female humans. And so an ovule is a structure in the seed plants, but in other organisms too, right? That contains inside of it, but in the plants, the ovule has inside of it a female gametophyte with an egg. And hence that gametophyte is better protected because it's inside of an ovule. And additionally, the ovule is made up of many different layers, which we'll talk about in this class today, named the nucellus and integuments. And so because the female gametophyte is inside in an ovule and surrounded by these multiple layers of tissue, she and her egg are better protected. Once the egg in the ovule becomes fertilized, then you will have the zygote, right? N egg and N sperm come together for 2N zygote. This is what's going to be inside of that seed. So when you have the ovule is a structure that has a female gametophyte and an egg inside. Once that egg is fertilized, you no longer have an ovule. You have a seed. So same structure, but different contents, right? And different things will happen converting the ovule into a seed. And right, so one thing, so whereas in the ovule there was an egg, in a seed there is a fertilized egg which grows into an embryo. And that is the result of egg and sperm coming together in the process of fertilization. All right, so this is a picture of an ovule. And I think this is from your book. What did I just say on the previous slide, right? What's inside of an ovule? A female gametophyte with an egg, and it's surrounded by different layers. So do I draw on it? Okay. These are the basic parts of any ovule, the basic parts. There will be different variations later on, but for right now, this is basic ovule anatomy. So I said on the previous, sorry, all my boxes are in the way here, right? An ovule is a structure that has inside of it female gametophyte and an egg, right? Okay, here is an ovule. So inside of that, remember, this is an extreme form of heterospory. So we have to have we have to have sporangia and we have to have spores still. Let's see, I think black still works. So this whole structure here is an ovule. Inside of the ovule, you have the sporangium. Because we're dealing with egg, because we're dealing with egg, we know there's female gametophyte. Sorry, I'm getting ahead of myself here. Let's slow down. We need the megasporangium to make the megaspore, and the megaspore will be what grows into the female gametophyte plant, right? Okay, so that's where we should start. But these are the basic parts. You have a megasporangium, which is this whole, this whole beige circle is the megasporangium. Can't really draw around it. AKA nucellus. In the ovule, the megasporangium is also called the nucellus. Yeah, which you could also see in your book. That megasporangium is going to give rise to spores, right? Megaspores. But when we get to the ovule, only one spore will survive, and that spore we call the functional megaspore. So that's what's shown here in red in the middle. And then surrounding that is these layers. So I think I go forward here, right? So here, megasporangium, aka nucellus, is the inner part of the ovule. It's shown in beige on this picture. And it's going to be what grows and ultimately will house the megaspore, functional megaspore. Functional megaspore is inside of that megasporangium, and that's what's ultimately going to give rise to the female gametophyte plant, right? Via mitosis and cytokinesis. And that female gametophyte plant is also going to stay inside of the nucellus, inside of the megasporangium. Surrounding the megasporangium, aka nucellus, are these layers of tissue, which are called the integument or integuments, depending on what we're talking about. Some things only have one layer of tissue for the integument, other things have more than one layer. But this is going to be serving that protection surrounding the megasporangium, really protecting ultimately the female gametophyte and her egg. Later on, we won't go into too much now, but later on, when the egg gets fertilized and the ovule starts to turn into a seed, it's going to be the integument that will turn into the seed coat, but we'll cover that more in detail later. And then remember, this functional megaspore here is going to grow into the female gametophyte plant, right? Spores grow into gametophyte plants. So that female gametophyte plant will have an egg on it, and we need sperm to be able to reach that egg. And so there's a hole at the top of the ovule called the micropile, and that is the hole that allows sperm to enter and ultimately fertilize the egg once the egg is there. Right now in this picture, there is no egg, right? Because the megaspore hasn't grown into a gametophyte plant yet. Okay. This is important. This is important. Make a note of this. Draw a star, highlight it right here. Make a note of this right here. So the megaspore, the megaspore is no longer released from the megasporangium. The megaspore stays inside, right? That's what I was saying here. Megaspore stays inside of the megasporangium, does not get released anymore, stays inside and turns into a female gametophyte plant. This is a big deal. We haven't seen this yet, right? We haven't seen where the megaspore stays inside the megasporangium, period, right? We haven't seen that before. What did we see before? Before we saw this, look, recall from Selaginella. Before, I'm gonna change colors. We saw this. I'll use purple. Look, Here is a strobolus of Selaginella. See, Selaginella. This is a strobolus of Selaginella. What's on a strobolus? Leaves that have sporangia on them, right? That's what's in a strobolus. Because we're talking about Selaginella, which is heterosporous, we know we're going to have ultimately two types of spores. So if we have two types of spores, we have to have two types of sporangia, and they're going to be on two types of leaves. They're really the same leaves, but they have different things on them, so we call them different names. So on this strobolus from Selaginella, because we are talking about something that's heterosporous, we have Megasporogenesis here and Microsporogenesis here, right? This whole thing that I'm circling is the Megasporogenesis. This whole thing that I'm circling is the Microsporogenesis. The Microsporogenesis is going to make, via meiosis, microspores. The Megasporogenesis is going to make, via meiosis, megaspores. But what happened next, right? What happened next? The microspores and the megaspores would be released out into the wind, right? The megaspore was released when we looked at Selaginella. But not anymore. In the seed plants, not anymore. Megaspore never gets released, stays inside. So when you're looking at these slides later, this is showing you the Megasporogenesis, and this is showing you the megaspore being released, right? Back when we were talking about Selaginella. Okay, remember? Here it goes again. Where was the female gametophyte plant back then, right? If this is seeming unfamiliar to you, go back and watch the recorded lecture, or look at the slides again, or look at your book again, or look at your notes again, or look at your lab material again. But back with Selaginella, that megaspore got released inside of the megaspore back then is where the female gametophyte plant was. And for what it's worth, sure, the gametophyte plant didn't really get released. The gametophyte plant stayed inside of the spore, but the spore got released, right? Which again is not what is happening in the seed plants anymore. The megaspore doesn't get released anymore. Megaspore is going to stay inside. But back with Selaginella, the megaspore got released. Female gametophyte plant lived inside, which is still the case here in the seed plants. The female gametophyte plant still going to live inside of the megaspore. But it's different. It's different than what we're going to see in the seed plants, because in the seed plants, what? What? Does the megaspore get released in the seed plants? No, no. Make a note of this. This is important. Okay. All right. So, as I said, so first, okay, so we're gonna, there's a few other things that are different. We're gonna look at them a little bit more closely. I think, yeah, good, okay. So one thing that's new is a significant event. See the title of the slide, significant events. One significant event is that the megaspores are no longer released and that they stay inside the megasporogenesis. That's one significant event in the evolution of the seed plants. Another significant event of the evolution of the seed plants is that there's only going to be one megaspore. Only one megaspore now inside of each megasporogenesis, right? So see here, there's only one megaspore mother cell, right? Or you could say megasporocyte, either one is fine, but there's only one of those inside the megasporogenesis. And when this megasporocyte or megaspore mother cell, which is diploid, goes through the process of meiosis, right? The result is going to initially be 4 megaspores because you can't, if you have a spore mother cell, and if what I'm about to say right now is confusing or sounds unfamiliar to you, go back to lecture 2 about alternation of generations and go back to that slide that I showed you with the roadmap of alternation of generations. If what I say right now is confusing in the next couple minutes, pause this and go and get that slide that I highlighted in the past about how important that one slide is. But you have the spore mother cell, right? In this case, megaspore mother cell. That's going to undergo meiosis. The result will be, because what's the result of meiosis? You should know this already. So I'm not going to tell you, you go back to lecture 2 if you don't know what meiosis results in. But here we have 4 megaspores initially, but 3 of them will die and only 1 survives. Sorry, it's like very dramatic. But yes, only 1 megaspore will survive, and that's what we call the functional megaspore. So we can finish that off, right? 1 functional megaspore. So what did we see before? What did we see back in Selaginella? When we looked at Selaginella, look, inside of the megasporangium, there were 4 megaspores per megasporangium. You can't see the 4th one in this slide, meaning this is a cross section through a strobolus. And the way that the strobolus is cut, you can't see the 4th megaspore because it's behind the other 2. But trust me, there were 4 in each of these megasporangia back in Selaginella. Whereas now there's only going to be one megaspore per megasporangium, aka functional megaspore. So just to sum up here, what are some significant events in the evolution of seed plants, right? One thing is that there's megaspores are no longer released. That's a significant event. Another one is that there's only going to be 1 megaspore per megasporangium. We call that the functional megaspore. Okay, how do we get to that functional megaspore? I'm going to show you how. So the way that you get to that functional megaspore is through a process called Megasporogenesis. And don't let the word scare you, because you should know by now that you can pull these words apart, right? Sporo, what do you think sporo refers to? It refers to spores, right? Megaspores, what does megaspore refer to, right? These spores that are going to grow the female gametophyte plant, because spores grow gametophyte plants. Genesis, we've already come across in this class, not today's class, but in previous ones, means the generation of, right? Or the beginning of. And so Megasporogenesis is the process which is going to generate megaspore. And we only have 1 functional one at the end. Okay, so this here, these drawings here are ovules. And I think, get rid of this thing. Yep, so I'm going to walk you through this process called Megasporogenesis. It's a process. So what happens? First thing is you have 1 sporocyte, which is a megasporocyte or megaspore mother cell. That's what I was talking about here, right? Only 1 megasporocyte, aka megaspore mother cell per megasporangium. That's what we see here. So there it is in the middle, that little dot in the middle. I'll use blue on this slide just to mix up the colors. right? This little dot is your sporocyte or spore mother cell, same thing. And it is diploid, and it's inside of this ovule. Look at those, you can see the parts we already covered, right? This large thing inside here is the megasporangium. These layers on the outside are the integument. And the dot in the middle is a spore mother cell or a sporocyte, megasporocyte. That cell, because it's one cell, which is diploid, is going to go through the process of meiosis 1 and 2. And the result, well, I guess I do tell you about meiosis here again. Good. And the result of meiosis 1 and 2 will be 4 haploid daughter cells, right? Because when you have meiosis, you go from a diploid thing to a haploid thing, and you go from 1 cell to, or 1 nuclei to 4 cells or 4 nuclei. Here they are in the middle. But you may notice, right, 4 haploid daughter cells. Here are the 4 of them right there. It's already, meiosis already happened. But you might notice that the one on the bottom is much bigger than the other 3 above it. And that's because of this here, unequal cytokinesis. So when meiosis occurs in this spore mother cell or megasporocyte or megaspore mother cell, same things. When meiosis occurs and cytokinesis follows, not all of the cellular, not all of the products are going to be distributed equally, right? One of the spores will get most of it. We'll get most of it. The other ones will be kind of puny. And that is going to be what is your functional megaspore, and it's going to grow into the female gametophyte plant. So I'm going to use colors really quick here. So just to get you oriented. So this is an integument, the outside of the ovule. This is the integument, the outside of the ovule. This is the integument, the outside of the ovule. This here is a spore mother cell, diploid. This here are, these here are the 4 haploid daughter cells that resulted from meiosis. Those are the megaspores. And one of them has most of the stuff. And so that's going to be your functional megaspore. The big one, this functional megaspore, is going to undergo, it's going to grow into the female gametophyte plant via mitosis and cytokinesis. So the first thing that that megaspore will do, or sorry, I should also do this. So this, I'll use a different color. I'll use yellow. I'll use blue. This whole thing is the megasporangium. This whole thing inside is the megasporangium. This whole, it's hard to draw. This thicker line in here, oops, is the megasporangium. Go back to our functional megaspore, which is here, right in the middle, the bigger one. That's going to undergo mitosis and cytokinesis and grow into the female gametophyte plant. And in this drawing here, female gametophyte plant, that's all this now, because that spore grew into the female gametophyte plant. Okay. Right. It's just showing you the functional megaspore. And it's showing you the female gametophyte plant inside of the megasporangium. Okay. On this female gametophyte plant, right? On this here, on this female gametophyte plant that I'm drawing in green, there is an egg, right? There's actually 2 eggs, but the female gametophyte plant has eggs on it because what does a gametophyte plant make? Gametes, egg and sperm. And if you're a female gametophyte plant, you make egg. And so after the egg is fertilized, that's going to develop once the egg is fertilized by a sperm, you have a zygote, which is diploid. The zygote will continue to grow via mitosis and cytokinesis into an embryo. And the embryo is going to develop where the egg was, which is where? On that female gametophyte plant, right? Because that's where the egg was. Remember, egg, you should be saying it out loud. Egg stays, right? So that's where the embryo will grow. As the embryo grows, the integument is surrounding, remember the integument back on this, right? So as the embryo grows, the integument is surrounding all of this. Oh, I think I'm telling you about sperm. Yeah, right. But yeah, sorry. So egg gets fertilized, embryo begins to develop. The integument is going to be protecting that area and will ultimately become the seed coat, right? Because sorry, I'm getting confused. Yes, so this should actually, I think, be on 2 different slides, because this is just saying, okay, the egg is fertilized and the embryo grows inside of where the female gametophyte plant is, right? Because she had the egg. Now we're going to talk a little bit about the structure of the ovule. So the ovule has the integument surrounding it completely, except at that micropile apex, right? Where sperm enters. Here, I'll just go back real quick, right? There's a hole here called the micropile where sperm will enter, right? Or where, yeah, same thing there. How did we get that structure? Why is there a hole there? Because a hole doesn't give a lot of protection, but we need the sperm, the microspores, to get to the egg. So we'll talk about that in a few slides. But here is that, so just to keep you oriented, this is where we left off on this slide. On the right, we had the female gametophyte plant inside of this ovule. Sperm fertilizes the egg on that gametophyte plant, and now it'll make a 2N zygote, which will grow into an embryo. That's what we see here. And now we don't call it an ovule anymore, we call it a seed, because we don't have an egg anymore, we have a baby plant. So to use the same colors I used before, here is where the integument was. Now it will become a seed coat. Let's see, I forget what color I used for well, I know what color I used for the gametophyte. So all of this here, the female gametophyte plant, right? She had an egg on her. She's still here inside of that megasporangium, which will also turn into seed stuff. But most importantly, what's in the middle, this whole area here, is the new embryo, right? Notice diploid, because it's the result of egg and sperm. Notice gametophyte still haploid, right? Yeah. Okay. Now I think we're going to talk a bit about, yes. So in terms of how the ovule evolved, we don't know the exact order of events because the fossil record is incomplete. So we have to make kind of some best guesses. We know that the seed plants occurred pretty early in the life of the vascular plants. So we do have evidence of ovules and seeds going back to around 365 million years ago. So here's our best as to how the ovule came about. So what we're going to cover right now is similar to when we discuss things like how enations turned into microphyls, or how megaphylls were the result of like branching structures that underwent different changes in morphology. This is the same idea here. We're going to talk about what we think happened over evolutionary time. So initially, when we do see early ovules in the fossil record, they look kind of like this. So that's one ovule that I just circled. And one thing you might notice is it's not completely closed in. It's not completely closed in. Like you have these almost little like finger-like things, but they're not fused together like this. It's kind of like this, right? It's not like this. Whereas modern day ovules are like this. They're completely fused. They're completely fused. But these early ones, 365 million years ago, they're more open, is the point. And what we think happened, see, so that's why I used orange on this, because you see here, lobes of integument. So like each of these little finger-like things are basically what will become the integument, but it's not completely closed 365 million years ago. So what we think happened is, just like we saw for that evolution of the megaphyll, that word overtopping. So we think that some branches here, like, so some of these grew super fast, like these, and some of these didn't grow so fast. And clear drawings. And as that happened, when the faster growing ones were sterile, like they didn't have any sporangia on them to grow into male or female gametophyte plants, right? Obviously here female gametophyte plants, because we're talking about ovule, but parts of the branches were sterile and grew fast. Other parts of the branches were fertile in that they had an ovule on it and grew slower. And over evolutionary time, and we see this in the fossil record, basically the lobes of the integument will kind of fuse together to completely close the ovule except the hole of the micropile. So this is like similar to what was shown on the previous slide. These like finger-like things, lobes of integument. Ultimately, those finger-like things probably like started to come together, but they still maybe weren't completely together. And then over time, maybe there's some slight adjustments, but the end result, so this is them starting to come together. Now the integument is like more fused, but the top is still really open. And then ultimately here, this whole thing is the ovule, this is the integument, and then here's that micropile hole for sperm to swim, or for sperm to enter, not swim. Yeah, that's the micropile. Right. So this is just an introduction to ovule, because we're going to be talking about seed plants, and all seed plants bear ovules. So that's why all species of seed plants bear ovules. So that's why we're going to talk about it. And as I mentioned at the top of the class, the seed and the ovule, because you can't have a seed without an ovule, are a huge evolutionary leap. And part of the reason that is because it gives the female gametophyte plant with an egg more protection, and then later the embryo more protection when you have the seed. And because this is such a successful way to protect the egg of the female gametophyte and the baby of the embryo, right, that is inside of the seed, the majority of plants on Earth are seed plants. So This is a pie chart we saw the very first day of class, I think. And you can see the green part of the pie is the seed plants. So the majority of the plants on Earth are seed plants. Hence, it's a very big advantage to have those structures. For the rest of this class, meaning the rest of this course, we're going to talk about all of the different types of seed plants that are so angiosperms and gymnosperms. We will start with the gymnosperms, which are not as abundant as the angiosperms. So if we go back here, if we were to take the green part of this pie and only look at those 377,000 species, so only looking at that green part, this is what would be inside of the green part. And you can see over 360,000 of those species are angiosperms. And only about 11,000, give or take, are gymnosperms. So we're going to talk about gymnosperms first in this course. But before we kind of go into them in more detail next classes, I want you to know what the terms mean, because we're going to see the terms gymnosperm and angiosperm many, many times. So you should know what it means. This word gymno comes from the Greek, which means naked, and sperma, which in Greek refers to a seed. So the gymnosperms are what are called plants that have naked seeds. If you have a naked seed, though, that means you also had naked ovules. So the ovules, and then later, so remember, I know this is new today and every class has new information, and I apologize for that, but that's how courses go. But here, so the ovule, remember, the ovule has in it a female gametophyte plant with an egg, right? But eventually the ovule gets fertilized, and then it is not called an ovule anymore, it's called a seed. So that's what I mean here when I say the ovules and later seeds are not enclosed in an ovary. So for example, today, when we went through these things, right, all of these things today, we never said the word ovary, right? We're just talking about an ovule. So no ovary. No ovary is the more basic type of seed, and hence naked seed, naked ovule, naked seed. So in gymnosperms, the ovules, gymnosperms, the ovules, and later the seeds are not enclosed in an ovary. In other words, they are naked. That means that if you're looking at a gymnosperm, you can see with your eyeballs the ovules, and after the egg inside of the ovule gets fertilized, you can see with your eyeballs the seeds, because they are visible, they are exposed to the environment. Anytime something is exposed to the environment, it's probably not as good as being protected, right? Hence why there's only 11,000 some odd gymnosperms, because they have ovules and seeds that are naked or exposed to the environment. Only 11,000 of those, whereas angiosperms, there's 366,000 of those, and that's because they provide better protection. So the word angiosperm, angion or angio means vessel or a container. Sperma still refers to seed. And so an angiosperm plant has ovules and later seeds that are enclosed in an ovary, meaning the ovules and later seeds are not exposed to the environment. which is a better system, obviously, because there's, excuse me, there's 366,000 angiosperms, whereas there's only 11,000 some odd gymnosperms. How do I get rid of those lines? Here we go. All right. Who are we going to be talking about next time? Oh, yeah. So just if you were to, I'm talking here on this slide about this word ovary, right? Naked seeds or gymnosperms have ovules and later seeds that are not enclosed in an ovary, whereas angiosperms have ovules and later seeds that are enclosed in an ovary. So just so you can see that on a drawing, here you go. So on the left is a gymnosperm ovule. See, look, this is the integument. Inside of it, same structures we talked about before. But notice that the ovule is not inside. So this whole thing here, sorry, I should use a different color, not the, this whole thing here is an ovule. And notice it's not inside of anything. It's naked. It's exposed to the environment because it's a gymnosperm. However, look at the angiosperm ovule, which is here. And what do you know? It's inside of something. In this slide, that something is pink. And what that something is the ovary. So in the angiosperms, the ovule is not exposed to the environment. It's protected and inside the ovary. Okay. So who are we going to meet next time? So next time we're going to learn about cycads, which are members of those gymnosperms. So cycads have naked ovules and naked seeds, right? So cycads will have the situation shown here on the left because they are gymnosperms. We'll talk about them next time. The class after that, we will talk about Ginkgo, which is shown here, which is also a gymnosperm. So hence naked seeds. And then in subsequent classes, we'll talk about conifers, which are also gymnosperms, naked seeds. And then after we finish all the gymnosperms, we will go into the angiosperms. So next class, we're going to talk about cycads, right? Cycads. And I think that's it. Yep. So with that, I hope you guys, let's see. I hope you guys have a great rest of your day. For those of you who emailed me about meeting for office hours, I'll get back to you later today as well. Yeah, if anyone else has any questions or concerns, feel free to email me. Otherwise, I will see you on Thursday. So, oh, and so just a reminder that this coming Monday, you're doing the Fern Lab. Fern Lab has been available on Canvas for a while now, so hopefully make sure you, before then, download it, read it, and from now on in lab, you will be asked to show your completed lab manual before leaving lab because we really want you guys to do better on the next lab exam. And so for that, we need to kind of keep track of your work a little more closely, I guess. All right, guys, keep going. Work hard. Bye.